Good morning. Welcome to rdhblueprint.ca. My name is David. Today we're getting ready for the board exam and we're looking at microbes and embryology. This will probably not be a very heavy course on the board exam. It might be one question or two, but would you like to know what those are? Probably, right? I can't predict what's going to be on the board exam in terms of some random orofacial fact or embryology or what's in a cell. But you might have one or two questions that are just on random facts of this kind of nature. So we're going to go over it. I'm going to go over it pretty quickly because you should already know this. You can review this on your own. I'm just highlighting what's the most important things. Let's chalk and talk. So chapter 1, let's go over a cell. You should know a cell. You should know what's in a cell, right? So the cell membrane, this is where we attack most bacteria, most pathogens, whether it's a fungi or bacteria. We're attacking the cell wall. If we can attack the cell wall, everything spills out. Think of your own skin. If you get stabbed with a knife, everything spills out. That's what we're doing. So we have a nucleus. We're going to look over ribosomes and mitochondria. How you're going to remember this is the cell membrane, that's like your skin. So it keeps everything in. It allows things to come in and out. Nucleus, that's the brain. That's the organelle that organizes the whole cell. Ribosomes, protein synthesis sites. Okay, that's where we make proteins. And mitochondria, it's the powerhouse of the cell. You should have a little diagram of a cell. I don't have one here for you. You can draw one yourself. Know what the basic things are. In case to get you a question like, what creates energy in a cell? This is grade 10 science, but it's not maybe on the board exam. You don't know. So nucleus is like the brain. That's how you're going to remember it. It stores the DNA, right? Ribosomes are made in the nucleolus, sorry. It's inside the nucleolus. It's like a smaller factory within the control center. It makes ribosomes, which are needed to build proteins throughout the cell. So without the nucleolus, cells could make ribosomes to produce proteins. So within the nucleus, you have a smaller thing called the nucleolus. What are ribosomes? They are protein factories. They are protein-making machines. They read instructions from your DNA and they build things. Right? Here's a bunch of people building. All right, you imagine each of these are ribosomes. They're all getting instructions of what's the pattern going to be, and they build. Ribosomes can float freely in the cell, or they can attach to the rough endoplasmic reticulum. Why is it called rough? Because it looks rough, because it's got a bunch of ribosomes on it. Rough endoplasmic reticulum, rough ER. Ribosomes are attached to its surface. Right? You can imagine these things are little ribosomes, and this is the ER there, endoplasmic reticulum. Modifies proteins after they've been made, folds proteins in the right shape, transport the proteins to the parts of the cell. So rough, think ribosomes. If you have a question what ER has ribosomes attached, it's the rough ER. The smooth ER, on the other hand, it makes lipids, fat. Okay, lipids, fats are smooth. They're slippery. They're needed for the cell membrane. So the cell membrane is going to get its lipids, because it's a biphosphorylate lipid layer. It's going to get that from the smooth ER. It also detoxifies. It breaks down harmful substances, and it stores calcium. So smooth ER has no ribosomes, and it handles lipids and detoxifies. The Golgi apparatus is like the post office. It receives things from the rough ER. It modifies them. It sorts them and then ships them out, either within the cell or to the membrane. Mitochondria, this is the powerhouse of the cell. Remember, you can have more than one of these organelles. A cell that's growing a lot might have many mitochondria. So when ameloblasts or odontoblasts are making a lot of dentin or enamel, there can be more mitochondria. Okay, they need more energy. They make ATP. They take food and make ATP. Adenosine triphosphate, that is what gasoline is for a car. ATP is for the body. And while we get gasoline from an oil refinery, we get ATP from the mitochondria. So it takes sugar, oxygen, mixed it into ATP, and water, and carbon dioxide. Without mitochondria, we'd have no energy. Lysosomes, they're the recycling center. Like the garbage disposal and recycling. Lice like to cut in half, right? It splits things in half. The waste removal, they destroy bacteria and viruses that enter. They can break them in half too, and they recycle. They break down larger things into smaller things that can be used somewhere else in the cell. Peroxisomes, they're detoxification specialists. They break down fatty acids and they detoxify harmful substances. Think of it this way. Peroxisomes are like a water treatment plant. They clean up dangerous chemicals to make them safer. Vacuoles, you think of it like this, just contain something. They store water, helps them maintain their shape. They can store nutrients. They can store waste products. And they can store ions or molecules like sodium, right? They can store things to be used later, right? That's what we have. We're storing our fruit to keep it from spoiling, and we can use it later on in the cell. Plant cells have one large central vacuole that takes up most of the cell, whereas animal cells have several small vacuoles. Cytoskeleton, again, this is what the cell's skeleton, it's the hard parts that make up, give it its structure. There's microtubules. These are the largest fibers. Microfilaments, they're smaller fibers. They help with moving things in the cell and the shape changes, and intermediate filaments, medium-sized. So these things give structure and help transport things within the cell. Centrioles, these are cell division organizers found only in animal cells. They organize division and they work in pairs. So they're going to go on opposite sides of the cell when we get to PMAT, which again is grade 10, grade 11 science. You should know this. Cell membrane is the gatekeeper. It's selectively permeable, which means it can choose what goes in and out. Who is coming in today, who is staying out. Protects you and keeps you safe. Communication, it receives signals from other cells, and it maintains homeostasis, the same status. Phospholipid bilayer. Remember this? You have phosphate group and two lipid heads, two lipid tails. That's what we're looking at. Plant-only organelles, they have a cell wall. So not only that they have a cell membrane, they also have a cell wall. It gives it a nice structure. It's made of cellulose and helps the plant stand rigid. The skeleton's on the outside. Remember, animal cells don't have a cell wall, only a cell membrane. That's the main difference. Plant-only organelles, chloroplasts, right? They're the solar panels of the plant. They capture sunlight through photosynthesis and make energy. That's how they get their energy. So they take carbon dioxide, water, and sunlight, and they make glucose and oxygen. Oxygen is released into the air, that's what we breathe, and they get the glucose they need for energy. Only cell plants and some bacteria have chloroplasts. We do not have chloroplasts. We can't make our own food from sunlight, can we? So remember, the nucleus is the brain, mitochondria is the powerhouse, Golgi apparatus is the post office, lysosome is the garbage disposal, and ribosomes are protein factories. There's a little summary chart for you. Let's look at cell transport. How are things moved in and out of the cell? There's passive, no energy required, no batteries required, and active, which means it requires ATP, adenosine triphosphate. So this is like rolling a ball downhill for passive. It doesn't take any extra energy, does it? You just let go and it rolls. Substances move from an area of high concentration to low concentration. That's passive, no power needed. All passive transport works that way. All active transport will work from a high concentration to low. Just like you put a whole bag of sugar or salt in one end of your swimming pool, and then you go measure the water in the other end of the pool, well, guess what? It's going to get some salt and sugar in it, isn't it? That's passive transport. Energy required, we're now moving from low concentration to high concentration. We're going uphill. It takes energy to move a ball uphill, doesn't it? So types of passive would be diffusion, osmosis, and facilitated diffusion. So diffusion, that's simply putting salt in water and it goes to the other side, right? Osmosis, now we're in the movement not of substances but water. So you have a sponge and you pour water on half of it, it's going to move to the other half of the sponge, isn't it? So that's passive. And facilitated diffusion, it gets some help to move, but it doesn't require ATP. Well, active, that's going to be a sodium-potassium pump, where potassium's going in and sodium's going out, or sodium's going out and potassium's coming in, and it's doing that to decrease or increase the level of sodium in the cell. Endocytosis or exocytosis, basically endo to take within the cell and exo to take outside of the cell. So here's the basic concept of diffusion. It's the movement of molecules from an area of high concentration to low. So we start with many molecules in one area, then they spread out randomly until we have basically the same amount through the whole cup of water. And we know that with salt, right? You put it in, it's going to spread out evenly, or sugar. When you spray perfume in the corner of a room, soon the whole room smells like perfume, doesn't it? It spreads out throughout the room without a fan on. Diffusion through a cell membrane. Small molecules like oxygen, carbon dioxide can do that. Lipid-soluble substances. So lipid-soluble means it can go through fats. Fat solution, like it can dissolve into fat. And some water molecules can go through. This is the difference between concentration between two areas. It's called the diffusion gradient. The steeper the gradient, the bigger the difference, the faster the diffusion occurs. And that makes sense. The drier the sponge is on one side, the more quickly the water is going to spread to that side, isn't it? Whereas if we were to have a wet sponge and I put a drop of food coloring on one half of it, it's not going to spread as fast to the other side. Or if the air is very humid or already full of a lot of water, it can't hold a lot of perfume. So you spray it and there's nowhere to go. So it may not spread to the other side of the room as well. But if the room is really dry, if you're in a desert and there's not much water in that air, and you spray perfume, you now put some moisture in the air, it's going to spread very quickly to the other side of the room. So it stops when both concentrations are equal on both sides. Now there's no movement. Osmosis is now water transport. It's not salt or sugar or another molecule that's transporting, it's water. It refers to water through a semi-permeable membrane, and it's going to move from a low solute concentration. So there's more water to an area of high solute concentration. That means there's less water. Moving from a place where there's less salt to where there's more. That's why we put salt on ice. What happens? The water goes into the salt, doesn't it? It melts. Water moves to dilute concentration gradients, solutions. If one side has more dissolved substances like salt, water will move to the side with salt. Water follows salt. That's a rule of the body. If we want to change how much water is in the body, we want to move salt. Okay, let's go over osmotic pressure. This is water across a membrane. The pressure of water that wants to cross that membrane. So if you have a thin membrane, say like a cell wall, okay, stay in your lungs, maybe a little like a cellular wall there, and it's dry on one side, but wet on the other, there's pressure there through osmosis that the water wants to go across that membrane. Or if there's a lot of salt on one side but not on the other, the water's going to want to go to the salt, and it will cross that membrane. That's what we're talking about. The greater the concentration of solutes, so salt or something solid in that water, the greater the osmotic pressure, the greater the pull is for that water to cross that membrane. And we need to maintain balance. This gets into left-sided and right-sided heart failure, that if you have water being backed up into the lungs area, so the water can't drain out of the pulmonary veins, then what happens is you have more blood sitting there by the lungs, and because the lungs are dry, the osmotic pressure brings water from the blood into the lungs, and they start spitting up pink spit because that's blood in the lungs. That's been by osmotic pressure because the water has backed up, the blood has backed up into the pulmonary system. You can also see why osmotic pressure is important for just the cells inside our body. If there's too much salt water or too many solutes around part of a cell, the water will actually burst out of the cell. Let's talk about that. That's the tonicity or tonic solutions. There's hypertonic, hypotonic, and isotonic. So hypertonic means, when we're talking about tonic, we're talking about the concentration of dissolved substances like salt inside the water. So hypotonic means more solute than what you're comparing to. Hypotonic solution means as less solute than we're comparing to. So in a hypertonic solution, there's more salt around the cell. So then what happens is water leaks out of the cell and it gets like this. You get kind of these little edges, kind of spiky. Isotonic, like isosceles triangle, iso is the same. So everything is the same. The amount of salt inside the cell is the same. Outside the cell, there's no net movement. Hypotonic, now there is less salt outside of the cell than there is inside. So then what happens is water from outside the cell comes in to try and reach equilibrium, so the cell is going to get larger. So isotonic solutions have a perfect balance. That's what we want a lot of solutions to be. That's saline solutions, trying to have the amount of salt that's same in your blood or your tears. Equal concentration. There's no net movement of water, so a 0.9% salt solution is used in saline medicine because it matches our body fluid concentration. So normal saline rinse is isotonic within your mouth. It won't cause irritation because it matches your natural fluid salt water balance. So when we're talking about hypertonic solutions, remember, we're always talking about how much salt or solutes does something have compared to the cell we just put in it. Hypertonic means it has more salt than the cell. Hypotonic means less salt than the cell. So hypertonic solution has a higher salt concentration than the cell or tissue you're putting in it. So then water is going to move out of that tissue into that salt. So the cell will shrink, won't it? If we put your skin, if we were to take off a finger and put in really salt water, all the water is going to move out of your cell, right? It's only the skin that keeps water inside of us. But if it wasn't for that skin there, the water would move through that membrane and we would shrink. That's called crenation. Salt water rinse. So half a teaspoon of salt water per cup of water. It's a hypertonic solution. If the board exam asks you after procedural and there's a lot of bleeding, should you give a hypotonic solution or a hypertonic solution? We want hypertonic. We want more salt. Why? So that the fluid in the bacteria is going to leave the bacteria and it will go into the solution and they'll dry out and die. Now, more of that water in your cheeks, of course your mouth is going to dry out a little bit too, but that's okay. You've got lots of water in you. Okay, your cheeks can rehydrate. The bacteria cannot, so they will die. Hypotonic solutions, this is when we have less salt than the cell. So it has almost no salt. So then water will move into the cell from the hypotonic solution. So it will swell. There's no, you can think of it this way, okay? It's starting to swell and it's going to burst. It gets bigger. There's a risk of lysis. It may burst. So if you were to drink completely sterile water, distilled, there's no salt in it, and you have that too much, that's dangerous for you. You need some salt in your body. This is dangerous for cells because they can burst. Plain water is hypotonic to cells, which is why we use saline, not plain water, for medical procedures. So here's a little summary chart. This is all online for you. Memorize it, make your own. It's a very basic understanding of what's going on. So saltwater rinse, we want to create a hypertonic solution. When osmosis occurs, water moves out of the bacterial cell walls into the saline rinse, and they dehydrate and shrink and die. And we decrease our risk for infection. Now, active transport, working against the flow. This requires ATP. Sometimes we need to move things across their natural flow. For example, nerve cells need to maintain high potassium inside and high sodium outside, the opposite of what would happen naturally. So you're going to need ATP to do that. The sodium-potassium pump. Grade 11. I remember this. So it is the most important example of active transport within our body. It happens the most often. It keeps the balance of sodium, which is Na+, and potassium, K+, that nerve and muscle need to function. So nerve cells, you remember, will start off as polarized, then we depolarize them, and now it becomes equal. Then we need to repolarize after, so the nerve can be fired again. So 3 sodiums out, 2 potassiums in. We need 1 ATP to do that, and that creates an electrical charge for the nerve signals. Sugar absorption. Here's another ATP in action, right? Simple sugar molecules like glucose, fructose, and galactose, these are the smallest sugar units. We'll get that into the nutrition. You should know what those are. Active absorptions. These sugars are absorbed through the intestines using active transport. So we absorb monosaccharides through active transport. It allows complete absorption of the sugars from food for energy, even when the gut cells already have plenty. So they already have lots of sugar, so it's not going to be diffusion or osmosis, but we want to get more. So we need ATP to do that. So we're going against the concentration gradient. All right, last bit, phagocytosis. That's to eat. So like dysphagia, difficulty eating. This one cell is eating another. Okay, that's what's going to happen. So how does it work? There's 4 stages. First, it detects it. So the cell detects, oh, I can tell there's something around here I don't belong, I'm going to follow it. So it recognizes the particles such as bacteria or cellular debris through specific receptors on its surface, right? It senses it, oh, I see you. You shouldn't be here. Then it extends its cell membrane outside. It's like it puts it in its mouth. Pseudopods, so little feet, like fake feet, go around it. That's what pods means, feet. Then close it. Then there's engulfment. It's fully inside the cell now. Then digestion. Lysosomes and organelles break it down and eat it. That's how we're fighting many bacteria and diseases in our body. So specialized white blood cells will do this, such as the macrophages, or large eater, and neutrophils. They will also do this. They are primary phagocytes. Remember, the first responders in a battle in our body are neutrophils. This is the critical way to get rid of pathogens, is phagocytosis. That's mainly how our cells are doing it. Pinocytosis, now we're drinking. Cell drinking. Unlike phagocytosis, which engulfs, now we're bringing liquid in. It's mainly to get water in, not to attack. It's not an immune system response. Pockets fill with extracellular fluid. It pinches off to form fine vesicles, and they bring fluid in and nutrients into the cell. So phagocytosis is eating solids, so mainly like foreign invaders to kill them. Pinocytosis, that's maintaining, we need more water nutrients in the cell. All right, that's Microbes and Embryology Part 1. There's several in this series. Go online, do the test, and move on. Don't go over this chapter again and again and again. It's not that important. It is worth going over at least once. Take care. Bye.